Hello everyone, how are you all? I hope you are all good. So in the last video, we discussed about cell and its components, okay? But in today's video, we will be discussing some organelles. And those organelles are none other than endoplasmic reticulum, Golgi apparatus, lysosomes, and secretory vesicles. So today, we will be discussing about endoplasmic reticulum, Golgi apparatus, lysosomes, and secretory vesicles. Okay, so first, endoplasmic reticulum are of two types. Okay, one is rough endoplasmic reticulum, and the other is smooth endoplasmic reticulum. Okay. Now, what is the function of rough endoplasmic reticulum? The main function of rough endoplasmic reticulum is to synthesize proteins. Okay. The main function of rough endoplasmic reticulum is to synthesize proteins, whereas the main function of smooth endoplasmic reticulum is to synthesize lipids. Okay. Now, how this formation of protein takes place, and we will see how this protein gets processed through rough endoplasmic reticulum followed by Golgi apparatus to the targeted place. Okay, we will understand this one in a story format first. Okay, so we all know rice. We have eaten rice. It's very common to all. So for cultivating rice, we require raw materials, right? We require raw materials. This is so after getting raw materials, what we will do? We will cultivate rice. Cultivation of rice. Okay. After cultivation, we will get the product rice. Yes. Okay. So after the rice has been formed, now this rice will be packed in a sack, big sack, and will be carried by the delivery boy. Okay. This delivery boy will take that particular big sack of rice to the company. Like there are many companies of rice. Okay. Let's assume. Now what this company will do? This company will put some brand, will do some packaging. Okay. So we can say modification. After modification, the rice will reach to the consumer or customer, which is the target. Okay. So we all understood this story. First, we got the raw materials. After that, we cultivate the rice. We got the rice, and this rice has been carried by the delivery boy. And this delivery boy has given those particular sack of rice to the company where modification, that means packaging and branding, has been done. After those particular modification, it has reached to the customer for the seller. Okay. Now, This raw material is the DNA. In a scientific explanation, okay, and this cultivation of rice is the DNA to protein, and this rice is the protein, the formed protein. Okay, now this protein will be taken up by the this delivery boy is the endoplasmic reticulum. Okay, this delivery boy will give this particular rice to the company, and this company is the Golgi Apparatus. Okay, in Golgi Apparatus, protein modification will be taking place, and after protein has been modified finally, it will be reaching to the targeted place where protein is needed. Okay, now if we see in a scientific explanation, this was a story format explanation, okay, to make you understand clearly and to give you more interest. Okay, now if we see the actual scientific mechanism, okay, so this is a endoplasmic reticulum. In rough endoplasmic reticulum, why it is rough? Why the rough word comes in the rough endoplasmic reticulum? The rough word comes because of the ribosomes that are embedded in the outer part of the rough endoplasmic reticulum. So that's why it gives a rough type appearance to this endoplasmic reticulum. So that's why the name is called as rough endoplasmic reticulum. So these are the ribosomes that are embedded in the rough endoplasmic reticulum. Okay, this black color. Okay, this is just a rough diagram. And smooth endoplasmic reticulum means smooth type appearance in the outer surface. So there is no ribosomes embedded in the smooth endoplasmic reticulum. Okay, fine. So now you got the difference. Rough endoplasmic reticulum means ribosomes are embedded in the outer surface of the endoplasmic reticulum. So that's why it is known as rough endoplasmic reticulum. And in the smooth endoplasmic reticulum, there is nothing embedded in the outer surface. So that's why it gives a smooth type of appearance. Okay, fine. So in rough endoplasmic reticulum, ribosomes embedded, and in smooth endoplasmic reticulum, no ribosomes embedded. Fine, clear? Okay. So that was the difference of RER and SER, rough endoplasmic reticulum and smooth endoplasmic reticulum. Okay, fine. Now, generally, this DNA is present in the nucleus. Okay, and this DNA to protein formation takes place in two places. One is in the nucleus and other is in the ribosomes. Okay, fine. So nucleus we will be discussing in other video. Okay, we will discuss in the details. In here, just listen. So we got to know that protein synthesis takes place in the ribosome. Okay, the final step. So let's discuss the final step here. Okay, How DNA gets converted to protein, that one we will discuss in some other lecture. Okay. Actually, it happens in the nucleus. DNA gets converted to mRNA. Then mRNA gets excreted out via nuclear pore into the cytoplasm, and in the rough endoplasmic reticulum, ribosomes are present. In there, that mRNA gets converted to protein. So we will be focusing here only the final step. Okay. So the protein has been synthesized in the ribosome. Okay. So these are the proteins, the already synthesized protein. Okay. Now what happened? After getting synthesized, the protein gets entered into the rough endoplasmic reticulum. Okay. Then this embedded protein gets transported from rough endoplasmic reticulum to smooth endoplasmic reticulum. Okay. This is rough endoplasmic reticulum, and this is smooth endoplasmic reticulum. Okay. Now what happened when this protein reaches the last part of the smooth endoplasmic reticulum? This protein gets covered by the layer of this smooth endoplasmic reticulum, which finally breaks, okay, which finally breaks and convert into a transport vesicles. Okay, this is a transport vesicles. Okay, now these transport vesicles will get processed to other cell organelle, that is the Golgi apparatus. Okay, these transport vesicles will fuse the outer membrane of Golgi apparatus. In the transport vesicles already present the proteins. Okay, when it gets fused in the outer membrane of the Golgi apparatus, these fused proteins enter into the Golgi apparatus. Okay, and after that, what happens? It gets fused with the outer membrane, and here also the other type of vesicles that is formed. Okay, and this vesicle is named as secretory vesicles. And when this protein enters into the Golgi apparatus, this protein gets modified, and the protein that is present in the secretory vesicles are the modified proteins. Okay. Now these modified proteins will go to the outer membrane of the cell via exocytosis. Okay. This is the outer membrane of the cell. Sorry, outer membrane of the cell. Okay. Via exocytosis, reaching to the target place. Okay. Fine. So these vesicles, secretory vesicles that has been formed by the Golgi apparatus. Okay. These vesicles can be formed other like lysosomes, Okay, fine. So lysosomes have some different properties, like it digests damaged cellular structures, digest food particles ingested by cells, and digest bacteria and other microorganisms. Okay, so these are some functions of lysosomes. So those vesicles can be get converted from the Golgi apparatus either to secretory vesicles or to the lysosomes. Okay, secretory vesicles mainly functions as the transportation, like the modified protein which gets modified inside the Golgi apparatus. After that, these secretory vesicles carry this modified protein and via exocytosis from the outer membrane of the cell reaching to the targeted place. Okay, so understood this complete process. Revising it again, the protein which has been formed in the ribosome gets entered into the rough endoplasmic reticulum. Then slowly it gets transported into the smooth endoplasmic reticulum, where little portion of the smooth endoplasmic reticulum gets breaked out, containing the protein, and that transport vesicles transports this protein to the Golgi apparatus by fusing the vesicle with the Golgi apparatus. Okay. Then that particular protein gets entered into the Golgi apparatus where it gets further modified, and that modified protein is been further transported via secretory vesicles where the outer membrane of the Golgi apparatus gets break down and form secretory vesicles, and that secretory vesicles get attached to the outer layer of the cell membrane, and via exocytosis it reaches to the targeted place. Okay, fine. And that vesicles which has been formed by the Golgi apparatus gets either converted to secretory vesicles or gets converted to lysosomes. Lysosomes have different functions, that is, number one, digest damaged cell structures, number two, digest food particles ingested by the cells, and the last, digest bacteria or other microorganisms which are harmful. Okay, to keep. So similarly, like protein transformation, how the protein gets transported from the rough endoplasmic reticulum, smooth endoplasmic reticulum to the targeted place, followed by the Golgi apparatus, and also by the transport and the secretory vesicles. Likewise, lipid also gets transported in the same way. Okay, so lipid also gets transported in a same way like protein transformation. But the only difference is lipid synthesis takes place in the smooth endoplasmic reticulum and protein synthesis takes place in the rough endoplasmic reticulum. Okay, but apart from that, the synthesized lipid, synthesized lipid, which is being synthesized in the smooth endoplasmic reticulum, gets incorporated in the smooth endoplasmic reticulum for the extensive growth. incorporated. So the synthesis lipids, which is being synthesized in the smooth endoplasmic reticulum, gets incorporated into the smooth endoplasmic reticulum itself for the extensive growth. And to maintain the growth, so that it doesn't grow beyond the cell requirement, so the lipid gets transported in the same way like protein transformation. Okay. Okay, fine. So we got to know the functions of endoplasmic reticulum, Golgi apparatus, lysosomes, secretory vesicles. Okay, fine. So if you have any doubt, you can ask me in the comment box. And it's a very easy process. By the story format, I have make you understood. Even though if you have any doubt, let me know. And in the next part, we will be discussing the other organelles like nucleus, mitochondria, ribosomes. So these are some organelles which we will be discussing in the next part.